utilizing the most recent technology. So that's why, so we are very happy today to hear such type of very effective this subject. I request Dr. Arup Mukherjee, the Journal President, to brief about the biography of Dr. Kutubuddin.
In fact, it is a pleasure to me to introduce Dr. Kutub, who joined in NRI in 2016, and he's an excellent scientist. He first posted at NBPGR, New Delhi. As long as I know him, he is young in age, but he is very mature in knowledge. And in fact, it is our proud privilege that we are getting an opportunity to listen to him on CRISPR-Cas tool in plant disease management. Dr. Kutub did his MSc and PhD from the most coveted Calcutta University. And after that, he joined ICR as a scientist, and he got a very coveted Fulbright postdoctoral fellowship, and he worked in Pennsylvania State University for 2 years on genome editing. He has published many research papers, excellent research papers in international journals like Nature Biotechnology, then Plant Biotechnology Journal, Plant Science, and many more. And in future also there are many more to come. And he has been awarded with many prestigious awards, as I told previously, like Fulbright postdoctoral fellowship. So now I invite Dr. Kutub to deliver his speech, and we are eagerly waiting to listen to you.
Okay, sir. Thank you very much for this invitation from Indian Phytopathological Society, especially the Secretary Dr. Gogoi, Joint Secretary Dr. Mondal, Professor Apurva Chaudhary, the next speaker, and obviously from our institute and this Eastern Journal chapter, as Dr. Mukherjee and Dr. Bag, and the host, Dr. Rajkumar. So it's my real pleasure to talk about genome editing and how this genome editing could be utilized for efficient plant disease management. So I'd be briefly going through the subject and then giving you nice examples how it is already being utilized throughout the world and how we could also utilize in our own scenario. So coming to my outline of today's presentation, it is divided into 4 different parts. In the part 1, I will be briefly introducing you what is genome editing and why it is needed. And then obviously we're talking about CRISPR-Cas9, very simplified way. And then in part 2, I will be talking about several tools and techniques with different case studies, like bacterial diseases, fungal diseases, viral diseases, and insect pest resistance. And part 3, I would be talking about a new emerging field, how CRISPR-Cas could be utilized in disease diagnosis. And in part 4, I would be talking about from my own experience working with single base editing. So we can do how CRISPR-Cas9 can also do changing a functional SNP from a base pair to a G base pair, from a C base pair to a T base pair. So this is the outline. So coming to, so whenever I go to introduce my slides, so I always keep this slide to refresh our idea about DNA and gene. So, you know, all the higher eukaryotes, mostly higher eukaryotes, they are nothing but a composed of trillion of cells like human body and also higher plants. So these cells, if you take an example of this is a plant cell and this has a nucleus. This nucleus is the head of this cell because this nucleus contains the genetic material which is called chromosome. And chromosome is nothing but the arrangement of a long thread that is called DNA and that is coiled around some protein. This protein is called histone protein. So to keep in mind in a simple way, you have to keep in mind that chromosome is nothing but a coiled thread of DNA. So if you see this DNA, the whole long thread of DNA is not that important, but some portion of that DNA is important. And this functional portion of a DNA is called a gene. And this gene is actually governs a character by expressing mRNA and protein. So a functional unit of DNA is called a gene, which actually governs a character. So now what is in our DNA? The DNA is nothing but the sequence of 4 different kind of nucleotides that we commonly know as nucleotide base. So these are 4 letters: adenine, guanine, cytosine, and thymine. So they make up our genetic code, and this genetic code actually governs almost everything in an organisms. So if you see, if I want to introduce you the genome editing, I should share you this slide with you. So this is a classical example of how plant and bacteria interact with each other. So if you see, this green box is a plant cell, is a rice cell, and when it is infected with, so you see, this rice has a special kind of gene. That gene is called sweet gene, and this gene actually produces some transporter protein. So I told you that one gene produces a protein to govern a character. So here in case sweet gene produces a transporter, this transporter protein is called sweet transporter, and that transporter is present on the cell membrane and cell wall. So if you see, what does these sweet transporter do? They transport sugar from inside the cell to the apoplastic region, up to the outside of the cell. This is their normal function. So now, why I'm talking to you about this sweet gene? Because this Xanthomonas oryzae is a very intelligent, pathogen. When Xanthomonas oryzae, they infect plant cell, rice cell, they secrete a protein, effector protein. This effector protein is called TAL effector. This is called transcription activator like effector. This is a protein, a small protein. So what this effector does, this effector is secreted from zoo, and it goes to bind to the promoter region of the sweet protein. So of the sweet gene. So any gene has in its upstream position one promoter. That promoter actually regulates its expression. So how much that gene is going to be expressed, that determines on the promoter sequence. So what this zoo actually does, it secretes this TAL effector protein, and this TAL effector goes to the promoter of sweet gene. So when it binds with the promoter of the sweet gene, then the sweet gene expresses in a very higher amount uh, uh, by this interference. So when it produces many number of the sweet protein, and they form this transporter in the cell membrane, then as a result, this cell is able to transport more sugar to the apoplastic region. So now in the apoplastic region, there are availability of more sugar. So what this zoo is doing like this, why? Because this zoo actually utilizes this sugar as their own nutrition. So if there is more nutrition, then zoo is very happy, and they will grow like anything because they are having access to their nutrition, okay? So they are utilizing plants' own gene to their own favor. So they are highly expressing the sweet gene so that more sweet protein produced, so that more sucrose get transported from inside to the outside one. So, and then you can see here a lot of zoo is getting replicated because of the availability of high amount of nutrition. So this is a classical example of how plant and pathogen interact with each other, and how they deploy each other's mechanism to their own benefit. So now the question is, we know this mechanism. We know that this TAL effector binds to the promoter sequence of the sweet protein. So now the question, if you see in details here, so this is the sweet gene, and this is the effector binding element. This is nothing but a sequence in the promoter, and with this sequence, this TAL effector binds. So now the question is, can we specifically disrupt the effector binding element in that promoter, that EBE sequence, so that the TAL effector will no longer be able to bind? And if it does not bind, then sweet gene would not be expressed in a higher amount. So the zoo would not be benefited. That's the hypothesis, okay? So the question is, we have to selectively disrupt that sequence in the promoter. So can we do that? The answer is yes. The way is yes. Why? This genome editing. So we can do that by genome editing. So there are 4 different well-known genome editing techniques: meganuclease, JDF, talin, and CRISPR-Cas9. So I'm not going into details of all earlier three, but I will be talking about CRISPR-Cas9 today. So, this meganuclease, JRFN, and TALEN, these three techniques, they depend on protein and DNA interaction. If you want to modify a DNA, if you want to modify a gene, then you have to go for protein DNA interaction. Means you have to design a new protein for changing a new DNA in each case. But in case of CRISPR-Cas9, it is not like that. So it depends on RNA and DNA interaction. So if you want to go for a new gene editing, then you have to design a new RNA because it depends on RNA DNA interaction. So you don't have to go for a new protein designing. So that's why CRISPR-Cas9 is easiest among these four, and it became very popular because of this simplified way of going from one gene to another, one gene to another. Okay, so yeah, this is let me share with you. This is Dan Bartas. Dan Bartas is the pioneer of talent technology. So I had met him in University of Minnesota. And this is Jennifer Doudna, who is the pioneer of CRISPR-Cas9 technology. And I met her in Cold Spring Harbor Laboratory. So yeah, coming to this technology, CRISPR-Cas9, this technology is actually revolutionizing agriculture, basic biology, and therapeutics in all the sense. So people are crazy about it because this CRISPR-Cas9 act as a Caesar, and it is a very simplified manner. Let me discuss with you how it actually works in genome editing. So it has two components. One is a customizable RNA that is called guide RNA, and another is a DNA cutting protein that's called Cas9. So if I want to give you an analogy, so this Cas9 protein is like a postman, and these guide RNA is like the address. It's like the genomic address. So if you design this guide RNA, so you are giving the genomic address to the postman. So the postman takes the genomic address, and now they will go to the genome to search where this address is present. It means where the similar sequences is present in the genome, because you have designed your own address, the guide RNA, okay? And this Cas9 take that guide RNA, form a complex, and it will go to the genome and search for the similar sequences as the RNA1 that you have given. So now they will search and bind to a similar DNA sequence. If they find similar DNA sequence in the genome, they will bind there and they will make a double strand break. So this Cas9 or this postman has two scissors in its hand. So with these two scissors, it will cut both the strand of a DNA. We know that DNA is a double strand structure. So one strand would be cut by one scissor, another strand would be cut by another scissor. So it makes a double strand break. So as a result, now we know that this Cas9 with the guide RNA, it can make a specific cut at the genome because you are designing your own guide RNA for a special gene, and then it will go to that gene and it will make a double strand break. Now, what happens when there is a double strand break in the genomic DNA? Because DNA is very important for any cellular organism. So cell takes it very seriously to repair this kind of break, because this is a very bad situation for cellular environment. So they try to repair immediately this kind of double strand break made by Cas9. So they go towards two different pathways. One is NHEJ, that is non-homologous end joining. Another is homology-directed repair. So among these two pathways, this NHEJ pathway is the more predominant one. So cell always go out of 100 times, almost 90 times, it repair the double strand break with this NHEJ pathway. But now, remember, this NHEJ pathway is a little bit error prone. So if they repeatedly repair this double strand break and they seal that end, they can insert some wrong nucleotide, wrong base, or delete some original bases. So because of this random insertion and deletion, because of its error proneness, the gene sequence become disrupted. Because this red is actually highlighting the random insertion or deletion. So because the open reading frame of that gene, the sequence of that gene is changed because of this new insertion or deletion from the original sequence, now this gene become non-functional because it would no longer produce the same protein as it should produce. Okay, so now this gene became non-functional. So you can do in a very easy way to disrupt or non-functionalize a gene of interest that you want to disrupt. Okay, so this is NHEJ, and this is mostly followed worldwide. Now another pathway is homology directed repair. This is extremely low in efficiency in eukaryotic cellular environment. But how it happens? For more precise editing, what they do in normal cellular condition, if they find a double strand break, then in any diploid organism or any polyploid organism, they have, suppose in case of any diploid organism, they have 2 copies of a single gene. So now suppose the Cas9 creates a double strand break in 1 copy of the gene, but the other copy of the gene is intact. Now what cell will do, they will see the intact copy as the template to repair the broken copy. Okay, so in cellular environment, the sister chromatid, they will see the sister chromatid that is not broken, and they will copy the same information in the broken end. So it is almost error-free repair, and that's called homology directed repair. Okay, so I'm not going into details of homology directed repair, but I will be going to give you example of a lot of this NHEJ mediated pathway and how it is being utilized in terms of plant pathology and all the different kind of applications. Okay, so this video would make it more easier for you to understand how actually CRISPR-Cas9 works.
Been adapted into a powerful tool for genomic research. There are 2 components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its 2 nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes.
Okay, so I think it's a bit more clear to you now. So now coming to my part 2 of this presentation. So in this part, I will be talking about how this CRISPR-Cas9 is being utilized in controlling plant diseases for different kind of pathogens. Like, let me start with bacterial pathogen. So I'd be giving a few good examples. I cannot encompass all the different examples that are available within 1 hour. So it is a huge task. So with this bacterial pathogen, let me start with the same example that I used to introduce you to the subject of genome editing. So, the host plant also have, we know that resistance gene actually confer resistance to any pathogen. We know that is called R gene, but there are the concept of S gene, that is called susceptibility gene. So what they do, they help in early pathogen establishment. They are present in the host plant, but they help pathogen for their establishment. They also can modulate the host defenses, means they can reduce the host defense. And also they can help the pathogen sustenance. As I have given you example that how SWIT11 gene actually helps bacterial pathogen for their own sustenance by providing more food to them. Okay, so that's an example of susceptibility gene that I have already given to you. So now if you see these that I talked to you, that if this EBE, that elicitor binding element, where the TAL effector from the bacteria binds and increases the expression of SWIT, this has been disrupted by using genome editing technology. So this is from Nature Biotech, from a collaboration. So broader spectrum resistance to bacterial bite in rice using genome editing, and also TALEN has been used. TALEN is another genome editing technology, has been used to disrupt this EBE sequence. And there are other at least 6 or 7 papers that describes successful deployment of genome editing to disrupt that sequence. And when this EBE sequence is disrupted, so the TAL effector can no longer bind with the sequence because it has been already changed. So TAL effector cannot recognize whether it is the sweet promoter or not. So they cannot bind. So sweet 11 is not being used by that pathogen. So the defense of rice plant get enhanced. So this gives broad spectrum resistance because if you disrupt more than one sweet gene in one go, so you can develop a broad spectrum resistance by using this technology. This is a very classical example of how genome editing could be utilized in a very rapidly to develop disease resistance against bacterial pathogens. So if I want to give you a snapshot of this one, so this is the wild type one that has not been edited, infected hugely with bacterial blight pathogen. Whether these are all genome edited lines, these are different genome edited lines, and you can see how restricted they are when it is edited. Okay. So now let me give you another example of bacteria. So this is another example for in citrus. So bacterial canker resistance. So here in case of citrus, this LOB1 gene is the susceptibility gene that is used by the bacterial pathogen of this disease. So they increase this LOB1 expression and they form these kind of pustules. And you can see if this is wild type, after 12 days of post-infection, this is hugely infected. The citrus leaf, further, these are the edited lines that has not shown that kind of effect due to this infection of that gene. So if this LOB promoter is disrupted with genome editing, so it is able to inhibit the pathogen infection, the bacterial infection. Similarly, this is a case for citrus. So now if you see the example of tomato, this has not been yet published, but this is a preprint from bioRxiv. So you see what they have found, this DMR6, this is downy mildew resistance 6, it is the ortholog gene present in tomato, and they have seen that when Pseudomonas syringae and other bacteria infect the tomato, then this gene gets very highly expressed. It means you can hypothesize that this gene is somehow either being utilized by the bacteria for their own purpose, for their own infection. So what they have done, trying to disrupt this DMR6 gene, and they have found significant reduction of disease incidence against this bacterial pathogen. So if you can identify any susceptibility gene and you can target to disrupt that gene function, and you can reduce the expression of that gene to inhibit or to develop resistance against that pathogen. So now coming to the fungal pathogen. So fungal pathogen, if we want to give you example, so this is also a case of susceptibility gene editing. This is mildew resistance locus, or MLO is a famous locus. Actually, that also act as a susceptibility locus. So in case of wheat, this wheat is a hexaploid one, so it has 6 copies of a single gene. Suppose if you want to see this Triticum aestivum MLO gene, so this gene has 6 copies in wheat genome. So now what they have done, they have selected a conserved region of that MLO gene that is conserved in all 6 copies of the gene. Okay, and then they targeted this gene with talin. Talin is another genome editing one. So, and they disrupted it. And if you see the result here, this is all hugely infected or infested with the powdery mildew pathogen, but not this one. What is this one? This one is the one which has edited all the copies. So you can see TAMLO, AA, BB, DD. So all 6 copies of that gene has been edited, and that line showing almost complete resistance against this powdery mildew fungus. Whether you can see this is the wild type, and these are the one. Suppose this is 2 copies of the gene edited, this is another 2 copies of the gene edited, this is 4 copies of the genes edited, 4 copies of the genes edited, and here is the 6 copies of the gene edited. So in case of wheat, a little bit difficult because all the 6 copies you have to edit. Otherwise, suppose 4 copies you have edited 4 copies, but the another 2 copies is still present, so pathogen can use that 2 copies for their own own benefit. So you have to disrupt all the 6 copies of the susceptibility gene. So you can see this is the wild type one, and this is the 6 copy edited of that MLO gene, and this has been published in Nature Biotech. So now this is the similar example of the same gene, orthologous gene in tomato, and it's a Solanum lycopersicum MLO gene, and it has been also edited in Solanum in tomato plant, and you can see this is the wild type panel, and this is the genome edited panel where it shows a great level of resistance against this pathogen. So now coming to this very crucial example of banana. You know, this banana is infected hugely with this Fusarium fungus that called tropical race 4 or TR4 strain. But these race of pathogen is devastating because, you know, recently Colombia in 2019 declares a national emergency as this TR4 pathogen disease has been confirmed in their banana industry. So, you know, let me give you an example. This Cavendish is one of the very widely marketed triploid species of Musa acuminata. You know, triploid means they are sterile in nature. They are not diploid or tetraploid, so they are not fertile. So you cannot improve the resistance power through traditional breeding because they are sterile. So you cannot do through improving through breeding. So you have to go for some other methods. So genome editing actually is a very attractive tool to edit this kind of triploid genome in this Cavendish banana. So this is a news published in Nature that CRISPR might be the banana's only hope against this deadly fungus, because the group of Lina 340 from International Institute of Tropical Agriculture from Kenya, they are working with that. So they have found some genes that are actually used by this pathogen for their own purpose. So they found that susceptibility gene, and they are going to disrupt that gene and to see how it affects to resist that Fusarium fungus TR4. So now coming to this deadly disease that is emerging disease, wheat blast, and I know my next lecture is on exclusively on wheat blast, so you'll be learning more about this disease in my next lecture. But let me say, just tell you a few sentences about this disease. So this Magnoporthia oryzae pathogenesis is a devastating fungus, as we all know. Why it is devastating? Because it is very rapidly, it could infect within 1 week, it can deform the grain of wheat. So actually farmers would not have any means to resist it if it is not counted within that week. So it is very rapid. So that's why it is really devastating to us, especially in Bangladesh. This is hugely infected, and now it is coming to our, because we are the border of the Bangladesh, so it can. I think North Bengal is also experiencing that problem. So you know, this is also being approached by a group of scientists from UK. You know, Nicholas Talbot is a big guy in plant pathology. So, and Sofian Kamun from Sainsbury Lab, and they collaborated with the Bangladesh scientists, and they visited Bangladesh, and they are now trying to develop novel blast-resistant wheat varieties for Bangladesh by genome editing. So what they are doing, what is their approach? You know, there are some susceptibility genes to rice blast disease. We know that rice blast disease is Magnoporthia oryzae. So there are some susceptibility genes present in rice. So when those susceptibility genes are mutated, then they lead to disease resistance. And what they are doing, they have they are going for bioinformatic analysis to find or identify putative orthologs of those susceptibility gene in wheat. So we know the gene, susceptibility gene in case of rice, and they are finding the similar kind of gene in wheat, and they found 10 susceptibility genes. It is from bioinformatic analysis. And now they are trying to modify these genes using CRISPR-Cas editing. So they will soon be probably publishing the result, but this is undergoing experiment that I have shared with you. So this is now coming to a different portion, not to only increase the host resistance power by editing the susceptibility genes, but also to understand the pathogenesis in a better manner so that we can figure out what would be the approach to actually going for developing a good management practice. So here, understanding pathogenesis, you can see this papain is a cysteine protease, and that helps in plant defense that is present in papaya plant. But they mostly have very immense role in plant defense. But this Phytophthora palmivora is a pathogen, is a fungal pathogen that could infect the papaya plant. This study is actually from Hawaii in America. So Hawaii is known for very good papaya production, you know. So what they have found that this pathogen actually has some cysteine protease inhibitors. I told you that cysteine protease plays important role in plant defense. Now, what this pathogen do, they have some inhibitor protein against this cysteine protease. So they have some inhibitor protein to inhibit the plant defense, okay? So now what they have done, they have found 5 putative inhibitors. Among them, these inhibitor, this EPIC8, when they mutated that inhibitor in the fungus, in Phytophthora palmivora, and what they have seen that the pathogen lost its virulence. So pathogen can no longer infect the papaya plant. So this is the actually virulence gene of that pathogen. So you can see here is the wild type one, this left one is the wild type one, and this is the one that's mutated fungus. This is the wild type one, this is mutated fungus, wild type one, this is mutated fungus, and you can see very reduced amount of infection. So now you know that what are the tools of that pathogen that they use to infect the plant. So you have a better understanding, and you can figure out what to do to inhibit that pathogen. So this is all a list of different pathogens. So genome editing is not only used to edit the plant gene to develop host resistance, but also to understand the pathogenesis. There are a lot of studies that describe genome editing in fungus, so that you can select, suppose you selected 10 putative genes that is likely to play in virulence. So you knock out one by one all of them, and then you can know that which gene is the most important to the pathogen for the pathogenesis mechanism. Okay, so this is the all fungus. So now coming to viral pathogen, this is an interesting case that I want to share with you. So you can develop an artificial immune system in your own plant of interest. So what I told you, that if you give the Cas9 your genomic address, then Cas9 takes the genomic address like the guide RNA, and then it goes and find the similarity with the DNA in the genome, and it can cut that genome. So now what if you give the Cas9 in your genome of interest? Like suppose this is the case of tobacco and Arabidopsis. Suppose in tobacco you transform Cas9 protein, and then you give an guide RNA which is actually similar to the viral DNA. We know that viral already, or most of the virus is sequenced, okay? So we know viral sequence and what are the portion of the viral genome that is important for their own replication. So if you design guide RNA against the viral genome, not against the plant genome. So plant genome has Cas9 and then guide RNA. This guide RNA is similar to the viral DNA. So now whenever a virus infects the plant cell, then this Cas9 and the guide RNA, they form complex, and they go and find the viral DNA. You know, whenever a virus infects a plant, they insert their genomic material into the plant cell. So whenever the DNA or RNA is coming, then this Cas9 go and bind with that viral DNA, so that, and the viral DNA is chopped out. So now you can degrade the viral genomic DNA. So the virus would no longer be able to replicate within the plant cell, so that the plant cell has an immune system against that virus. So this has been published in Nature Plant. So this is another approach has been used against tomato yellow leaf curl virus. However, they have used 2 guide RNA. One is against coat protein of that virus, another is the replicase 1. So these are the 2 important protein coding gene for the virus. So what they have done, they did Cas9 transformation and the guide RNA for disrupting the coat protein gene of that virus and the replicase gene of that virus, so that it can develop resistance against that virus. Okay. Now this is another interesting story of against turnip mosaic virus. a major pathogen in all the field-grown vegetable crops. So many viruses, they use host machinery to replicate themselves, to produce their own protein. Okay, so this is a classical example that some viruses, they use host, this is eukaryotic initiation factor 4E. This is a protein that helps in translating from mRNA to protein. So this helps to produce protein. So these gene is actually, this protein is used by the virus to translate viral protein. Okay, so now what they have done to mutate the plant, this gene in the plant, so that the virus cannot use the same protein for their own translation. So if viral protein is not produced, so the packaging would not happen. So the virus would no longer be formed, new particles. So the plant would be resistant, okay? So now coming to another good example from Lina Tripaঠি's group that I told you, they are working on banana. So see, this is the, this BSV virus, banana streak virus, is an example of Badna virus, and this Badna virus has a very peculiar mechanism. What they do, this Badna virus genome can integrate into the. It's normally found that there are two kind of banana. One is derived from A genome, that's Musa acuminata, another is from B genome, that's called Musa bulbiciana. So what they have, it is already, this basic study has been done, and they have found lot of Badna virus sequences is integrated into the B genome. So B genome is inherently susceptible to that virus because it contains viral genome, okay? So it's very difficult to breed that kind of B genome banana for disease resistance, okay? So this plantain is another very popular banana cultivar, plantain, and it has AAB. So B is a part of that plantain genome, but that B genome has already integrated BSV sequence. So what they have done, this Lina Tripaঠি's group, they have identified the sequences where they have integrated into the B genome of the banana, and they disrupted especially the viral genome that is integrated into the host genome. The host genome already contains the viral genome. Okay. Now this, especially the viral genome portion, is disrupted using CRISPR-Cas9, and it gives a very good result to overcome the major challenge of banana breeding. So you can see these are the 3 edited line, and this is the wild type edited line, wild type line that is not edited. So it gives a very good amount of resistance against that disease. So okay, sorry. So okay, so now you can ask me, okay, so you are all, yes, I have given you example of DNA virus, but there are a lot of viruses that has genomic material RNA, not DNA. So is there anything, so you might question me, so is there anything in the CRISPR-Cas toolbox that could be used against this kind of RNA virus? Yeah, the answer is yes, because this Cas13 is a very good orthodox of Cas9 protein that I told you. So this Cas13 is an orthodox protein. Orthodox is nothing but like, it's a brother protein of Cas9. And this protein has a unique property to go and bind RNA instead of DNA. Cas9 go and bind DNA and cut the DNA, but Cas13, they go and bind RNA and then cut and chop out that RNA. So this Cas13 could be used to develop resistance against this kind of RNA viruses. There's another from AFN Cas9. This is a species Francisella novicida, is a bacteria. This Cas9 also can disrupt RNA instead of DNA. So those could be used to develop resistance against RNA viruses. Yeah, this is an example of how this AFN Cas9 has been used to develop resistance against a RNA virus. So this is, you can see, this is the wild type one, and this is the one that has AFN Cas9 and guide RNA, and they became resistant to that kind of RNA viruses. So now coming to insect pests. So is there anything to develop insect pest resistance in plant? So this is an example of BPH resistance development. So, this BPH is attracted to rice plant, is known from the basic biological studies that it's for serotonin. So if more serotonin is produced, then BPH is more attracted towards the rice plant. So this serotonin production producing pathway, there is one gene. This gene is targeted by CRISPR-Cas9 to disrupt that gene and to reduce the production of serotonin so that the BPH is no longer attracted towards this plant. So this is the wild type and this is the mutant one. This wild type is heavily infested, whether the mutant is least infested with this one. So yeah, now understanding the pest. So now you have to understand the pest better. So this same technology CRISPR-Cas9 could be used to understand the pest. So this has been used. Whitefly is a very menace to agriculture because it is a pest and it carry different kind of viruses. So recently in CRISPR journal they have demonstrated successful editing of this whitefly genome. You can see this is the wild type one, the eye color, and this is the one that is edited. The eye color has been changed due to the editing of the white gene in that genome. Similarly, we know that brown plant hopper, this BPH, that is pest for many crops, especially rice. So you can see these two gene has been demonstrated to be successfully edited by CRISPR-Cas9, the cinnabar and white, and this eye color has been changed. This is the wild type eye color, and this is the edited one, the eye color has been changed. So this CRISPR-Cas9 is also working in insect genome. So yeah, if you want to know about more what are the crucial genes in that insect pest, so you can go for CRISPR-Cas9 to know better about this one. Okay, so this is another very interesting example, and World Health Organization is thinking seriously to apply this, especially in African countries, because this mosquito is a very dangerous enemy of human because it can spread all those kind of diseases. So this is called gene drive technology using CRISPR. So normal standard inheritance is if you want to mate these two insects through mosquito, one is mutated, another is not mutated. This is a wild type one. So the offspring have a chance of 50% of inheritance of that mutation. So the mutation actually goes through the generation and it's a very slow manner, okay? But what you can do, you can manipulate that inheritance, normal inheritance pattern by using CRISPR-Cas. So it's called gene drive. So it is gene drive containing mosquito when it is made with the wild type one. So nearly 100% of the offspring inherit the modified gene in almost 3 to 4 generations. All the population would carry that same mutation. So now the hypothesis here, if you generate a mutation that actually makes them sterile, they would no longer produce egg. That kind of mutation you can create with the gene drive cassette. And then if you allow them to mate with the wild type one, and this mutation goes in a vigorous manner, so in 4th or 5th generation, even in 7th generation, all the population would be sterile. So this has been how it is done actually. So you see, this is the 2 chromosomes. Suppose in a diploid insect, this is the 2 chromosome. One chromosome contains the gene drive. This is the red color is the new insertion using the CRISPR-Cas9, and it has all the mechanisms to copy the same sequences in the other chromosome. So in the same single generation, the other chromosome has also the same kind of mutation. So the both chromosome contains the same mutation in a single generation. So in the next one, both the gametes would contain the same gene drive that is lethal to their own egg production. Okay. So if you see, there are several papers of gene drive and a very good journal in Nature Biotech. So it is applied in Anopheles gambiae, double sex female gene drive they have done, and there is no egg production has been found in 7 to 11 generation, and total population collapsed in a caged population. It has been released, it has not been released in the wild, but it is being done in caged population. It has been found that within 7 to 11 generation, all the population become sterile, so they can no longer breed to produce more mosquito. So this is a very exciting field, especially to resist this kind of mosquito, especially in African countries. So now coming to the part 3 of my presentation, this is disease diagnosis. How CRISPR is helping in disease diagnosis. This is another kind of Cas protein, Cas12a, that has a property of, it is called collateral cutting. Collateral cutting means you have a specific binding, suppose Cas12a, you give them a guide RNA, it goes and binds with the target, and then it cuts the target. That's the normal mechanism. But when it finds the target one and bind, it cuts the target obviously, but it also can cut another DNA if it is present around this protein. Okay, so this has been used in detection. So what they have done, so this is the Cas12 one, Cas12 protein, this box, and this is the guide RNA for the Cas12. Suppose this guide RNA is targeted to a pathogen DNA. Okay, so if it founds the pathogen, then it would bind and cut the pathogen DNA. That's the normal things. But how it could be used in diagnosis? So in that same tube, if you use this kind of small DNA, which has a coenchain dye and a reporter dye, the yellow is the reporter dye and this is a coenchain dye. So if reporter and coenchain are both present, then the reporter would no longer fluoresce. But if coenchain is separated out, then reporter would give you fluorescence and you could detect it. Okay, so how it is done? Suppose in a tube, if you give Cas12a guide RNA, and if that tube contains the pathogen's DNA, then this guide RNA bind there and Cas12a cut that. And I said to you, Cas12a has collateral activity. So it can also cut this reporter coenchain 1. So if the reporter coenchain is separated by the activity of Cas12a, then this reporter actually fluoresces, and you would detect that this tube contains our pathogen DNA. So this sample is positive for the pathogen. So this is CRISPR detection. You can read more. This is one from Jennifer, another from Feng Zhang. This two method is very famous, detector and Sherlock. And it is being used in, approved by FDA to use for COVID-19 detection. CRISPR-Cas12 based detection of SARS-CoV-2. And, now it is being used for plant genes, and these are the two publications recently came. It could be used for trait detection, pest surveillance, GMO detection, or also pathogen identification. The same one in a very rapid manner and a cheap way. So this is diagnosis. Yeah, in my lab, he is my supervisor. His lab, just Matt Whitley, is recently graduated, got his degree. He was working on early detection of citrus greening pathogen. So this is being used in all over the world now, but not published yet too much for plant disease. But it is a very good area to explore. Now coming to single base editing, my work with single base editing. So I told you that CRISPR-Cas9 could do random indel, and it can deactivate a gene. But this is an example of the movie from Pa. So this is Amitabh Bachchan, and he died at his 13 because of infection, because of a disease called progeria. Progeria is a rare genetic disease, and she is healthy and he is affected with that disease. So why he is affected? Because of the presence of a nucleotide instead of a G nucleotide from the healthy personnel. So because of the single nucleotide polymorphism, he is affected with progeria disease and he dies at 13. This is an example of plant perspective. This Kitake is susceptible to a blast pathogen because it has a C in certain position of a gene. Whether this mutant has a T instead of C, so it becomes resistant because of the single nucleotide polymorphism. So now the question is there a way to rewrite genetic letters precisely from one letter to another? The answer is yes, because the point mutation could be also created using CRISPR base editor. And this is an example of adenine base editor, where you can use this base editor to convert adenine to guanine. And this cytosine base editor could be used from C to T. Okay, so I'm not going to into details. Okay, so you can see this video.
To transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene.
Okay, so what we have done, we have targeted two different loci in rice to see whether this is working in rice plant, whether we could precisely change one nucleotide to other. So two genes we have targeted, one is WSL5, another is Z3, and if I can show you this exciting result of from our research, that this is the control one. This is a sequencing chromatogram. So you see this green peak is designated the A nucleotide. So I targeted this A nucleotide to change it to a G nucleotide. Okay, this is the SNP that I want to change from A to G. So when we applied base editor to this kind of plant, then what we got excitingly, this A is precisely changed into a green peak, what is a G. And you can see the other peaks, they're all equal, only this green peak has been changed to a G. So this has been, because of these changes, we have seen these altered characteristics features. So this is white stripe leaf gene. So we have mutated this gene and we got this white stripe leaf phenotype. So because of this SNP changes, this kind of phenotypic changes we have found. Now for the second gene, Z3, the same, this A was our target to change it to a G, and this base editor could change precisely this A nucleotide to a G nucleotide. And all the other nucleotides has not been changed, they remain unchanged. And because of this single nucleotide change by base editor, we have found this kind of expected phenotype like light green, dark green, light green, dark green, light green. So this is a zebra 3 phenotype that we could achieve by applying base editor. So base editor free mutant has been obtained very quickly. So, let me give you an example. So suppose base editor, you transform base editor into the genome, and it is integrated into chromosome 1, and now your target is in chromosome 4. So this base editor construct would produce base editor protein, and it would go to target chromosome 4, and it would change that single nucleotide that you targeted to change. Okay, now if the target is changed, so you don't need this base editor any longer to present in your genome. So in the next generation, if you segregate them out and select only the target mutation but not the base editor, then you can achieve a homozygous targeted mutation but without any transgene integrated. So this is an example from our results. So it is an example of only 25 plants representative data. And out of 25, we could achieve in T1 generation, these 1, 2, 3, 4, 5, 6 plants did not contain any base editor construct by the absence of that gene. You can see this is the indication, but they have that mutation that we targeted for, that WSL5 and Z3. So you can achieve transgene-free mutant plant in a very rapid manner in the next generation itself. So this is a classical example that we targeted to develop a disease resistance rice variety. So this is an example of a gene that has a premature stop codon. This TAG is the signal to the translating machinery to terminate the translation or to terminate the protein synthesis. So in one genotype, because of the presence of a premature stop codon, this TAG, these gene cannot produce a whole length of protein, the full length of protein. So it became disease susceptible. Now, when we apply base editor, then we could change this TAG to TGG. You can see TGG has been changed, TAG to change to TGG by using base editor. So A has been changed to G. So now there is no stop codon premature. So this mutant would produce the full length of protein and it would be disease resistant. So this is still going on, this work. Okay, so yeah, to know more, if you want to read more about this base editing, you can go to our review article that we wrote for Trends in Biotech about different technical consideration and practical application. And this has been selected as the best review of from all the Trends journal for that issue. And we also wrote another article for predicting that CRISPR-Cas9 random indel, what kind of indel that would be generated by using CRISPR-Cas9, that you could consult with this one. And if you want to read the story of that zebra tree and WSL5, this is freely accessible, published one. And this is recently a protocol book that had been published, edited by us. So in order to make it easy for those who are new in this area, we have published this book where all the protocols is present to apply this technology to different kind of organisms, including fungal organisms, bacterial organisms, even algae. So yeah, and this is the blog that you would go for. It is written in a layman language about plant-based editors. So coming to conclusion of my presentation for part one and part two, what I've told, genome editing has unprecedented ability to generate targeted and specific mutations in almost every loci of plants, pest, and pathogen. And it is a great tool to study host pathogen and host pest interaction by modifying both the genome. And indeed, this amazing tool has great potential to provide alternative means to effectively manage crop diseases. In part three, I talked about diagnostic tool. CRISPR-Cas toolbox has a lot to offer. However, the field is not in its full bloom in case of plant disease diagnosis. So you could explore this one. In part four, what I've told, precise single base alteration is a wonderful tool for installing functional SNP for crop improvement. Thank you very much for giving me a chance to talk in this webinar, and thank you especially to Dr. Arup Mukherjee, Dr. Manish Bhatt for selecting me, and also to the Secretary IPS and Additional Secretary and all the hosts. Thank you very much.
Okay, thank you, Dr. Kutubuddin. It is very, very nice talk. It was going on, going on, going on. It is very prolonged, very nice. So you have covered every aspect, right from the bacterial, fungal, viral pathogen. Also, you have touched insect, even DNA viruses, RNA viruses. In case of insect, how fecundity can be challenged with the help of this CRISPR-Cas technology. So it was very informative talk for the viewers also, for us also. Since this is the time is almost over, so we have received many questions, but quickly we can have few question answer. So Dr. Kutub, can you chat the questions from your side?
Any two?
Any two, yeah.
Okay, so 1 second. Most of the chat I have seen very good lecture on all this kind of thing, but not any question.
There is one question and
yeah.
answer.
Oh, question answer. Okay, chat. Oh, Q&A. Okay, so good. How to identify a susceptibility gene for any disease? This is a question. So yeah, this susceptibility gene, if you want to understand, suppose if I want to give you a case study, but that's not the case for all diseases, but suppose you did one resistant genotypes and one susceptible genotypes. If you compare their transcriptome and if you see that the susceptible genotypes actually has few genes transcript is expressed very high amount, it means you can hypothesize from that data that the genotype is susceptible and this gene is expressing in a very high amount. So probably those genes is actually helping the pathogen. So you can hypothesize that, and then you select those highly expressed gene in the susceptible variety, and then you can go for CRISPR-Cas9 to disrupt them in the susceptible variety one by one and see their effect, whether they are able to increase the resistance power or not. If they are increasing resistance power, it means your hypothesis is correct that those genes, any of those genes are actually susceptible genes. Okay, this is one mechanism, but there are a lot, so I can answer those later. So yes, whether Cas tools can be used in plant virus disease management. I think it is the question from Binod Upadhyay. I think I have answered in my presentation that how Cas tools is being used for plant virus disease management. Whether Cas12 is used to identify the latent viral infection in plants. Yes, the Cas12 is being used. There are two papers that used Cas12 to use this viral infection in plants. But as I told you, this has not been used widely, at least from the published literature. But many, I know personally, many labs are trying to use this technique for viral infection or early detection of plant pathogen in the plant. to make a good measure before the onset of the symptom. Okay. So next question is, could you please give an idea about the current scenario of genome editing from plant disease management in India? So truly speaking, India is a very, very initial situation and a naive situation of application of genome editing for plant disease management, but many labs have started working on that, but not in a good pace as it should be in order to compete with the world scenario. But world has gone so far, but we are not at that level. Okay, so another question is.
Thank you, Dr. Kutubuddin.
Okay. Yeah.
So here we are very happy. So we are very happy that you delivered your nice talk. It is